Okay, a little bit late because I had some technical difficulties, but let's go ahead and get started. Let's start with a word prayer. Dear God, it's Friday, it's the end of the week, and I thank you very much for that. I'm sure the students here thank you very much for that. I pray that you help us get through this hour, learn the things that we need to learn, and get on to the business of the weekend. I thank you very much. Praise. Okay. Who's tea? It's almost like someone knew I wasn't going to have my tea this morning. Like, hey, let's do this here. Okay, so we began talking about lysosomes and peroxisomes last class period on Wednesday. We got through a few different things, but where I ended talking about was, when I talked about this, is Greek for hydrolytic particle. Wait. I'm recording, right? I don't remember if I actually started or not. Yeah, I'm recording. Okay, perfect. And they are smaller. They tend to be smaller than lysosomes. Or yeah, lysosomes are smaller than lysosomes. Oops. They tend to be, some lysosomes are smaller than other lysosomes. Okay, so they tend to be fairly small organelles. They're 0.2, 0.5 micrometers in diameter. And they contain somewhere in between 40 and 50 hydrolase enzymes. Again, this is stuff I covered yesterday, so I'm just zooming right through this really quickly. And not yesterday, Wednesday. And they tend to break down macromolecules. That's kind of the point here of these. Sometimes they're called the garbage disposals. I'm not going to go into a digression there. One other thing, they tend to have an optimal activity of all the enzymes in there at a very low pH, somewhere between 4.8 and 5.8 pH. So this is fairly low, fairly acidic, and to maintain this pH, we have to have a fairly active method for moving protons into the lysosome. So proton ATPases are an important transport that's on the membranes of these organelles. Okay, I think that's pretty much where I left off, so I'll start going slower. So really there are three main functions, or digestive functions, that lysosomes have. And the first is the degradation of endocytosed material. So when the cell endocytoses certain things, it wants to degrade them. This could be for several different reasons, because it's a waste and it needs to get rid of it, but also because we have a macromolecule that we want to turn to simpler things. So for instance, if we have a polysaccharide that we want to turn into monosaccharides and then use those monosaccharides for other things like tagging proteins or other stuff like that, that kind of degradation could be happening as well. Forget bacteria here for a minute. I'm not going to talk about anything related to bacteria endocytosis here. But because we're talking here about endocytosis, and we just talked about the creation of vesicles, we're going to talk a little bit about how endocytosis happens, one particular kind of endocytosis. And the first thing that tends to happen is we have on the exterior portion of the cell, so if we have cell here, we'll have some receptors for some certain macromolecules that will bind some macromolecule here. So like we have triangle macromolecule, because those are important macromolecules in the cell, the triangles, and that will bind to the receptor, and by that way we will aggregate some sort of macromolecule that we want to endocytose. The second part here is that on the interior side, we begin coating the membrane with clathrin. And that begins forming a vesicle like we would see otherwise by getting a small with our little clathrin Yeah, whatever. You know what clathrin looks like. And so, this is what's known as clathrin-mediated endocytosis. It is a subset of a larger type of endocytosis known as, that's a bad one. There we go. I'll throw you away, okay. Receptor-mediated endocytosis. So receptor-mediated because, again, here we have a receptor on the cell surface that is allowing for the aggregation of the molecules, or the recognition and aggregation of the molecules that we want to endocytose. And then the coding protein that we're using is clathrin, and so this subset of this is clathrin-mediated endocytosis. So after that, this vesicle is going to form the rest of it, as we've talked about slightly before. After the vesicle completely forms and separates itself from the membrane, we have the clathrin unbinds, or that clathrin coat falls off. And this is then the endocytic vesicle. We have endocytosed whatever it is we want. So now what we have is, instead of this, these, we have vesicle, and we have triangle in the vesicle. A couple other things that this could be called is an endosome, a phagosome. For this purposes, don't worry about phagosome. Phagosome is what's used when we talk about phagocytosis. And usually that is the endocytosis of something larger. It undergoes a different fundamental process than what we're talking about right here. But just know that a phagosome is also a vesicle of material that has been brought into the cell by this basic process. But when we're talking about receptor-mediated endocytosis, we're going to be talking about a thing called an endosome. And then it's only really called a vesicle after shedding the clathrin. It's kind of a nascent vesicle before that. So this kind of giving you an idea. There's a lot of details on here that you don't necessarily need to know, but we have receptors up here that are going to bind and accumulate some sort of molecule. We then get clathrin, begin forming or coating the membrane there. We start forming the vesicle, and then eventually it buds off of the rest of the membrane. The clathrin falls off, and then we have our endosome, which is our last kind of right here. So oftentimes we will want to degrade the things that are in there. So again, remember, if we want to bring in a bunch of polysaccharides and turn them into monosaccharides or something like that, we now have to break these down. And we're going to do that using a lysosome. So the lysosome, as it is initially created, we make this brand new little vesicle coming from the Golgi, and we've packed it full of all the nice enzymes that it wants and got it down to the proper pH, and it's all good and active. That is the primary lysosome. It's a lysosome that hasn't really started to do anything with its life yet. It's full of potential, but nothing has actually happened. And what will happen then is we can bind that with an endocytic vesicle, so that we're essentially dumping the contents of each of these vesicles into each other. So the lysosome contents are going to mix with the endocytic vesicle contents, and we get the reactions of any of those enzymes that are in the lysosome with whatever are the contents of the endosome, and this forms what we know as a secondary lysosome. So it's still a lysosome. It's just, starting to live up to its potential now. It's moved out of the house, and everything's happy because, the parents want to move out of the house and then bind with something. Anyway, so that's a secondary lysosome. However, on the test, if you say a secondary lysosome is a lysosome that moved out of the house, that's wrong. Yeah.
Is it one that gets a low?
What's that?
Is it one that gets a low?
One that gets a low? Well, yeah, it kind of, actually, that is a much better analogy than one I came up with. It's a loping with the endocytic vesicle. I like this. Very good, Ethan. Thank you for coming up with better analogies than I do. Okay. So, and then whatever wastes are in there or whatever macromolecules that we have in there get degraded by the enzymes that we have in the lysosome, and then those can be recycled, used in whatever sort of processes that we need. This can also be then resecreted from the cell, ejected from the cell somehow. There's several different fates of what happens there. Okay, so there's this kind of cool little pathway where you can take things from the outside of the cell and get them on the inside of the cell. And that's mostly good. Sometimes it's kind of bad because there are also a reasonable number of pathogens that hijack the clathrin-mediated endocytosis pathway. Friday, in speaking English, not good combinations. I don't know. So certain bacteria and viruses will actually invade the cell through this pathway by essentially mimicking or activating the receptors that are used to start the formation of this endocytic vesicle and then getting themselves endocytosed. I list a few bacteria that actually end up doing this. We have Listeria, which is not good. Yersinia. So Yersinia pestis is what causes the plague, the bubonic plague. So that's a bad thing. Salmonella, Neisseria, which causes gonorrhea and meningitis. So these are kind of bad things. Things I'd rather not have. On the other hand, viruses, influenza, something that many of you have probably had at some point, got into your cells by this way, so you can curse clathrin when you got the flu next time. Ebola, which is somewhat worse than influenza, SARS. Yeah.
It's way worse.
It's worse, but less infectious. So hooray. Because if Ebola was infectious as the flu, we would really be in a bad place. Okay.
Why is it infectious?
Because first off, it's not airborne, and so you there are some really bad ones, like whooping cough, whatever the name for whooping cough is. Like whooping cough, if you go into a room that had someone with whooping cough in it 2 hours previous, you can still catch whooping cough. It's crazy infectious because airborne, it actually survives really nicely in the air for a long time. Influenza, not nearly that bad. Ebola, you actually have to touch the fluids or get some fluids of someone that's infected inside you. So, coming in 2 hours after someone with Ebola's been there, no problem. Anyways, what's that? Yeah, or something like that. Hopefully they didn't do that. Okay, a couple other things that lysosomes do. For instance, like autophagy, which is the selective destruction of organelles or other sorts of intracellular components that are old, damaged, no longer needed. We need to get rid of those things because they're broken, don't work anymore. We talked a little bit about this when we were talking about selective destruction of rough endoplasmic reticulums. I don't know. What is the plural for reticula? Whatever. And so this is another function of lysosomes here. Now, it actually turns out that many, at least at the molecular level, we're having fairly high turnover rates of many of your organelles. I mean, on the order of days, not on the order of weeks or years. And so, for instance, the nuclei of all of your cells will probably be basically different next time I see you this time next week. Wait, that has no sense. By the time you come in Friday, most of your nuclei in your body are going to be different. Not actually that we are destroying an entire nuclei and creating a new one, but many of these, what we're talking about, especially with nuclei and plasma membranes, we're actually taking portions of them and recycling them, so the entire thing ends up getting recycled once every about a little less than a week. We also have mitochondria. Mitochondria undergo wholesale destruction. less than a week as well. So just slightly less than a week, all of your mitochondria are new mitochondria. Hey, new things. And cytosolic proteins actually end up being turned over at a pretty high rate as well. Plasma membranes get turned over really, really fast. This is mostly because these are really active. This is where most of the interface with other things in your body is happening, and you are constantly putting new lipids, new proteins, all these other stuff constantly into your membranes, and then taking chunks of them back out and recycling them. So your plasma membranes end up getting recycled very quickly. So, and again, so with many of these, we're actually individual components of it get recycled, and so we end up over a certain amount of time get recycling of the entire organelle. But some of them, like the mitochondria, tend to be like wholesale recycling as well. Now, we've talked about some of this, for instance, chaperone-mediated autophagy were individual proteins. And again, one thing we talked about, like for instance, some other sorts of protein turnover a little while ago, especially chaperone-mediated. Proteins can also be broken down by lysosomes as well. And so certain, again here, individual proteins can be tagged for destruction by a lysosome as well. So when we're thinking about protein destruction and protein recycling, it's not happening just by one pathway. We actually have a plethora of different pathways that are breaking down proteins and recycling those amino acids to be used again. And the lysosome is one of the organelles that's involved in that. Here's another thing I was talking about is microautophagy, which here isn't the entire organelle deciding that we need to destroy the entire organelle. We are just engulfing some small portion of it, and everything, all the molecules are there within, and then recycling that portion of an organelle. So in this way, we can have like the organelle as a whole structure be a constant whole structure, but over time, turn over all the molecules that'll end up making up that organelle. Also, sometimes what you can have is a breakage or something not working correctly in some certain portion of an organelle, and we can selectively recycle that portion of the organelle without entirely breaking down the entire organelle itself. And then we also have macroautophagy, in which the entire organelle is going to undergo program destruction. And so this is also can be a large portion of our cell membrane as well. So it could be some large portion. And in the beginning, what happens is the entire organelle or an entire portion of the membrane gets wrapped up in another double membrane. Which makes sense. If we're going to like blow something up, you kind of want to build walls around it so you don't blow everything else up as well. Okay, that one goes so well. So you want to separate it off so we can like wreak destruction this limited area without destroying lots of other things around it. We're not exactly sure where that double membrane comes from. It could be coming from the rough endoplasmic reticulum. That's our best estimation at this point, but we're not really sure. And then what we have here is what's known as the autophagosome. It's essentially where you have phagocytosed some specific area of yourself, essentially, and this is going to fuse with a primary lysosome and do essentially the same thing that we have seen before with the secondary lysosome of what we saw earlier with an endocytic vesicle. Don't worry about this last line. I forgot to take that out since last year. We talked a little bit more about this pathway. We're a little bit behind, so I'm not going to do that this year. Okay, so that is 3 of the different functions or the digestive functions of the lysosome. The final in that is we can digest extracellular material by actually having the lysosome, instead of endocytosing this material, bringing in and then binding a lysosome to that so that we can dump its contents into there and digest it internally. We can actually also bind the lysosome with the cell membrane of the cell and then secrete the enzymes that are inside of that out into the extracellular space, and then to digest some material in the extracellular space as well. And this can also be peripheral proteins or peripheral things on the plasma membrane as well. Are there any questions with that?
So, apoptosis is the destruction of the cell.
Yes.
It is the destruction of organelles in the cell.
Yes, components or organelles in the cell. Yeah. It doesn't have to necessarily be the entire organelle. It can be parts or other macromolecules and stuff. Yeah. Autophagy is going to be destruction of some limited portion of the cell.
So basically, when autophagy isn't good enough, then the cell is marked for destruction.
Yes. That's correct. I mean, so it's not necessarily in that this is a road that automatically leads down to that way that doesn't work, because autophagy is also like a normal regulatory function. It's not that autophagy leads there necessarily, but if you have breakdown of something systemic, and also you don't necessarily always start with limited autophagy and end up finding, or before you go to apoptosis, you can go straight to apoptosis. So, for instance, during development, oftentimes you just have signal profound cell death where this cell needs to die. Yep, exactly. And so you just go straight to apoptosis. Or other factors. That's the one that comes right to my head. There's other environmental factors in which you just go straight to apoptosis. Okay. Yeah, Ethan?
So I'm guessing that after the enzyme is secreted and break down, the material in the extracellular space, the enzymes are breaking down the
Something in the extracellular space.
okay, and then it's getting excreted to the cell or ejected to the cell afterwards, or where does it go?
It depends. It depends on a lot of different things. Some of the breakdown products then can be endocytosed itself into the cell. So you don't want the entire thing, but you want the breakdown products. So you expel enzymes into the extracellular space. It's going to break down things. Sometimes this happens, for instance, with large extracellular proteins that are really stable. For instance, like things like collagen and stuff like that. You don't want to. It's really difficult to like break down because collagens form these cross-linked networks that then it's really hard to pull up an individual molecule. But you need to remodel those collagen networks periodically, because the collagen then begins to break down and you start having cross-links breaking. So you need to, over time, be turning over those matrices. And so you can't just pull an entire collagen protein into the cell by itself, because it's part of this larger network and it's still well cross-linked. So what you do instead is that you dump lysosome enzymes out into the extracellular space. You have a lot of proteases in there that start breaking down the various proteins in that network. You endocytose those amino acids where you can remake new proteins, and then also start remodeling that extracellular network. Does that make sense?
Yeah, so we're basically breaking stuff down to
That's not the only thing that happens. Sometimes it stays in the extracellular space because it needed there. Sometimes it's things like waste, which then get pulled into the circulatory system, get transported other places where they can be ejected from the body as a whole. There's a lot of different things. Sometimes you need to just break down things on, especially really small animals, on the animal surface. So there's really a whole bunch of different pathways that broken down material can take once you've broken it down. But I say the simplest thing is it could stay outside the cell, either be taken away or just stay there, or it could be endocytosed back into the cell. Okay, make sense?
Yeah, and that, one more question.
Yeah.
So we've talked a lot about things getting secreted from the cells. And I know that obviously the circulatory system will take that other person throughout the body, waste to move it and stuff that's needed somewhere else, take where it to go. But so like there's millions of cells. So and they're all stacked up. So do they like pass it from cell to cell to get it?
It depends. So they're not
Every
always
cell's
stacked.
not touching a capillary or something.
It's not always necessarily right between each other. So you do have between tissues and within cells things like lymphatic fluid and other things like this. And this is kind of getting into like the animal phys end of things beyond, because now we're talking about entire tissues and entire organ systems working together. But a lot of these kind of just products that accumulate around cells that are broken down waste and stuff end up in the extracellular fluid. The extracellular fluid then generally collects, because you don't have like a tight packing of cells always or right together. You do have things like that where you need to have complete fluid in impermeability, like urinary bladders, because it'd be bad if urine leaked out of your bladder. Around blood vessels, around other things like this, you have very, very tight and controlled movement of fluids. But through most of your tissues, actually, you allow fluids to move through fairly readily. And so it can collect a lot of these things, and then it tends to aggregate into lymphatic ducts, and then into the lymphatic system, where then the fluid portion gets recycled, returned back into the blood, which then goes back out to tissues. It leaks out capillaries, and then it rebathes the cells again as the extracellular fluid. And the waste materials out of that tend to get filtered out, and then there's several destinations that you can have for essentially the filtered material from your the lymphatic fluid. This is also why your lymph nodes swell when you have an infection or something else like this, because one of the junk things that accumulates very much in those cells or in the extracellular fluid is a lot of breakdown products of viruses and bacteria and stuff that you are collecting as you're fighting off that infection. As that then works its way through the lymphatic system, you're separating out essentially the waste, solid, not necessarily solid, but the non-fluid waste products from the fluid that can then be returned to the back to the circulatory system. And then those waste products start accumulating in your lymph nodes, which is where essentially that processing takes place, and then you get swollen lymph nodes. Yeah. All right, any questions?
It's Levi's birthday.
Oh, yeah, it is! I didn't forget. He told me this before, and I totally forgot. It's Levi's birthday. Happy birthday to you. Woo! Happy birthday to you. Woo! Happy birthday, dear Levi. Happy birthday to you. How old are you?
I'm 21.
21?
Yeah.
So what are you doing to celebrate your birthday?
Obviously. I don't know, maybe bake some cookies.
I see. I'm gonna bake some cookies. That's a good idea. Exactly what I did on my 21st birthday. Any other plans? No, just the cookies. Enjoy those. I say bring some in, but So hey, you're 21. Which of course brings us to Peroxide. What's that?
That's a good transition.
Yeah, I thought so. Because who doesn't want peroxisome on your birthday? Okay, so we have peroxisomes that are somewhat similar to lysosomes with a couple of differences. First off, they tend to be really small. They're smaller than lysosomes are. In other words, saying that we're about smaller than 0.2 micrometers in diameter each of them, so very small little things. And they tend to have crystalline enzymes. You can form crystals out of organic macromolecules, that's kind of cool. And these tend to have enzymes that are crystallized, and you can actually see under microscopy these enzymatic crystals in them, and that's how people have identified them. Now, these don't tend to be everywhere or really abundant everywhere. Most cells have a few peroxisomes in them, although some cells tend to have many, many more peroxisomes. They tend to be closely associated in places where we have a lot of high metabolic activity, which we'll come back to in a little while. But in vertebrates, this means primarily we're having a lot in liver and then also in kidneys. These tend to be places of fairly high metabolic activity, and we'll come back to in a couple moments why they're there. But then also in the leaves and seeds of plants. Again, these are places of high amounts of energy conversion and energy usage and capturing in plants. Excuse me. Had too many cookies apparently. So these tend to be in the leaves and seeds of plants as well. And then also unicellular organisms have some as well. Because, they only have one cell, so they need it in that one cell. So, it seems like peroxisomes originate from the endoplasmic reticulum. So we're not necessarily going through the Golgi to create these things. However, they also seem to receive vesicles from the mitochondria as well. We're not exactly sure what those do. Actually, I haven't checked since last year. We might have figured out since last year what they do, but I didn't find anything good, by last year. So, and they do a couple. Did I go too fast? They come from the mitochondria. We don't know what they do. Well, they don't come. They get vesicles received to the peroxisomes from the mitochondria that add things to them, probably, but we're not quite sure what those are for. So they seem to have a few functions that, on the surface at least, seem to be fairly unrelated, but they tend to align with types of metabolic activity. Number one, we kind of talked about this a little bit before, but this is one of the primary locations in which we are mediating the effects of photorespiration in plants. So phosphoglycolate, after we turn it into a couple other things, eventually gets transported to the peroxisomes, in which we're going to carry out some more conversion of the breakdown products of phosphoglycolate, so we can eventually make it into 3-phosphoglycerate again. This is one of the major functions of a peroxisome. In plants, you might have noticed that you do not photorespire, so it's not a vertebrate function. Along with that, beta oxidation, particularly in plants and fungus. So beta oxidation, again, is the sequential removal of carbons from long-chain fatty acids and breaking those down for energy. And in this case, in animals, so this happens a lot in plants and fungi, and it also happens in animals, but when it happens in animals, we're talking about very long-chain fatty acids. They tend to be 22 carbon dioxide, or carbons and longer. Additionally, gluconeogenesis. So essentially using other things, non-carbohydrates, to make more glucose so that we can feed this into things that we need glucose for. So things like from fats, from amino acids, from lactate. This also tends to happen in peroxisomes. Again, this is a big function that's happening. You look location-wise in you, this is happening in your liver predominantly. This is one of the big reasons why you have an abundance of peroxisomes in your liver to carry out functions like this.
Why is lactate?
What's that?
Why is lactate?
Because lactate is really important for making. You have this thing known as lactate shuttle in your body. So what happens is that when you are, and this again is getting into a kind of an animal phys range of things because it's not involving a single cell, but when you were doing something like running or something else like this, what you do is you'll end up in the anaerobic parts, like we talked about earlier with anaerobic glycolysis, making lactate as your end product, so you can keep running glycolysis in the absence of oxygen, right? Now the great thing is that what you do is that not when you are undergoing anaerobic processes, your entire body is not undergoing anaerobic processes at the same level. Some places are more aerobic than other places. Does that make sense? So what you can do is you can move lactate from places that tend to be anaerobic to places that tend to be more aerobic. You can turn it back into glucose and their own glycolysis and everything else normally. And so this is actually really important processes that keep you alive when you're doing, active things. And so moving lactate around your body and then recovering the glucose from that, and actually you don't run it generally all the way back to glucose, it's usually intermediate, and then you run glycolysis forward so you can feed into Krebs cycle and all the rest. Another thing that happens is that now that's not what happens to all of your lactate. Some of it stays like in your fairly anaerobic places like muscles and other stuff like that. And once you have it, you need to recover that. So you actually use oxygen to take lactate, turn it back into glucose, and then oftentimes if you're fairly quiescent, go all the way back then to glycogen so that you can store it back again. And so that at some later point, that's going to cost you a few ATP to do that, but you got the ATP out, and now you're in like a quiet, okay place. It's okay to use ATP to restore that lactate back into glucose and glycogen so that at some future point, you can get like the whole 25 or so ATP you could have gotten from that instead of just completely wasting it. Does that make sense?
Yep.
Yeah.
And that's what the term gluconeogenesis is.
Well, yeah, gluconeogenesis is just making carbohydrates, particularly glucose, out of non-carbohydrate molecules. And then another function that, for which this is the first function that we knew these did, and this is what they yield their name from, peroxisomes, is the breaking down of reactive oxygen species, particularly oxygen-free radicals. And so reactive oxygen, just in general, oxygen is not good for organic molecules. It tends to react with things, break them down, kind of bad. And so this is especially true when we have oxygen that is in states that's reactive, and so we want to get rid of them somehow. Give you an example of a couple of these. Hydrogen peroxide, really reactive, breaks things down. This is why it's good for disinfecting, because it just kills things. Which is really weird. I knew a person once that thought, hey, or actually it was a group of people, there's like some kind of quacks out there at some point, it's like, hey, it's good for you to drink hydrogen peroxide. Yeah, that's not good. Anyways, don't do that. Don't drink hydrogen peroxide. And don't eat. Never mind. I won't say that. I was gonna say don't eat Fire Valley Nukes, but that's beside the point. Because that's tetrodotoxin. That's okay. Never mind. I'll save that next quarter. Hydroxyl radicals. You don't want these around, and then superoxide radicals as well. All of these bad, make lots of destruction, life unhappy. Peroxisomes, there's many like superoxide dismutase and a few other enzymes in peroxisomes that tend to break these down, and that is good. React them with something so that they are no longer in a reactive state, and they're no longer going to break things inside your cells. Another one besides superoxide dismutase is called catalase. Catalase breaks down specifically hydrogen peroxide. We react a couple of them together, and we get a couple waters out of that and an oxygen. And both of those are okay because, hey, that works well. Again, most of the reactive oxygen species that you make are going to be during energy consuming or energy capturing events. These often are occurring around kind of metabolic processes. So again, kind of continuing the general theme that peroxisomes are used abundantly during metabolic processes.
Stupid question.
Yes? Peroxisomes in red blood cells. I imagine probably not. Red blood cells have very, very few organelles.
There's like a deficiency in GPDH. We learned about this in my chem, whatever. And then that produces an ADPH which breaks down the peroxides. So we have peroxisomes.
Yeah, there might be, but yeah, in general, red blood cells are ametabolic and pretty organelle without many organelles. They may have a few, I'm not sure. Clea.
I was going to say, well, because the deficiency is affected by the fact that you don't have as much of the separate enzyme that wants to get rid of peroxide, so you wouldn't have peroxisomes in red blood cells because you wouldn't need that enzyme.
Yeah, that makes sense. Yeah. And whatever, here's another thing. It's like, this all kind of leads from, you have no nuclei in red blood cells. This makes it so you can give your capsules, all fun stuff like that. But if you have no nuclei, basically everything proteinaceous that follows from that you can't make. And those that you do will rapidly degrade. So, like you saw, organelles don't last very long in general. As soon as a red blood cell is made and finalized without it becomes a nucleate, it's destined to die because it can't maintain itself. And so, and this is particularly bad with very reactive organelles because you can't control them. And they tend to, if they start to break, they start to kill things. Like if you have Golgi and it's starting to break down and start releasing random enzymes and everything, that's bad. If you have mitochondria going and they start to break down, they tend to release lots of reactive oxygen because that's bad. So you don't really have mitochondria working. Okay, so a couple different things. Hydrogen peroxide bad. It oxidizes everything that's organic, particularly in you. That's bad in amino acids, nucleic acids, and these are most oxygen-free radicals are going to be byproducts of metabolic processes. Like for here in the mitochondria and the chloroplast during electron transport chain things, we are creating hydrogen peroxide. And again, we're going to damage like basically every organic molecule that you can get your hands on or they can get their hands on. So a couple different enzymes that are in there. One that we just talked about, catalase, also superoxide dismutase. These are good. Okay, I have like 2 slides left, but it's like at the time now. So I mean, it's Friday, and so I'm not going to keep you for 2 slides. I'll talk about them on Monday. And then we'll move on to the next whole thing, which is cytoskeleton. Yes.